Well, thank you, everyone. And thanks for coming out early in the morning. I'm Peter Kasson. I'm pretty new here. This was a picture from yesterday morning of the truck leaving Virginia with the freezers on their way down. So my lab is very much in the process of moving into IBB and getting going. So these should show up today or tomorrow, and some of her other equipment on Monday, and we're ready to get going. But we're interested in enveloped virus entry from a mechanistic viewpoint with an eye towards what are the evolutionary and sort of larger host pathogen consequences. So enveloped viruses have to cross a number of barriers in order to infect. Obviously, sometimes barriers between species. The envelope viruses have a membrane around them, and for the genome to be able to replicate in a cell, they undergo this process of membrane fusion to release the genome into the cytoplasm, which then, of course, we can study at very high resolution or lower resolution. And we're interested in questions about what are the physical constraints on viral entry, and also how viruses might evolve to cross host species or evolve in time for more efficient infection. So we're interested in what physically and mechanistically controls where in a cell a virus might infect. This is a schematic of a virus. We'll talk through this more later, but binding at the plasma membrane and going through the endocytic pathway. And different viruses enter at different points in this pathway, from the plasma membrane to very late in the lysosomal pathway. And figuring out what controls that. Also, figuring out what controls which host species a virus might be able to infect and transmit between. This is obviously something that's a great concern for emergence of pandemic pathogens, as well as other things. And so our goals really, apologies, I think that's actually the people with the freezers trying to deliver them. Our goals are to understand the biology that controls viral entry, to be able to assess pandemic risk of different pre-pandemic pathogens, and to be able to help evaluate countermeasures against potential pandemic pathogens. So we're really interested in a fairly zoomed-in view of viral infection, but with an eye towards what does this tell us about these much more macro scale behaviors and risks. The way that a lot of viruses get into cells, this is a schematic for influenza, which is a virus we've worked on for years, that is that it's sort of a prototype for envelope virus entry, and is also it's a proven, before 2020, I'd get up and say, this is the virus that has probably the best track record of causing human pandemics repeatedly. Obviously, now we have another major player, but that influenza virus binds to sialic acid terminated glycans on the cell surface. It's endocytosed, and acidification of the endosome triggers a conformational change in the coat protein that makes both influenza insert peptides into the host membrane and refold this protein to bring the two membranes together. And there are multiple key aspects here that these peptide membrane interactions are critical to the infectious outcomes. And we'll talk about this more later, that it's really not just a problem of the protein sort of squeeze the two membranes through, the genome pops out, and infection's done. That you can have point mutations to these peptides that can abrogate infection. And we'll also talk about critical properties of the host membrane. So we're really interested, this isn't the order we'll talk about it today, but about what's the role of binding to cellular receptors in the first place? What are the properties of the host membrane that control infection? And then, of course, what can we learn about, particularly in the protein membrane interactions, about activation and inhibition of the fusion machinery? So where I want to start is this question that influenza and many other viruses, dengue, chikungunya are some examples, infect primarily or exclusively via endosomes. We're interested in why. So this is an electron micrograph from some of our work showing an influenza virus inside an endosome. This is a single virus tracking trace from some colleagues' work where it's following the virus as it's endocytosed by the cell, and then the star is when it finally fuses. And the genome is able to enter and begin the process of replication. So there's been very long-standing work, I think originally shown by my colleague at UVA, Judy White, that influenza, although it goes through endosomes, you can exogenously make it fuse at the plasma membrane. If you bind influenza virus to cells and then essentially wash the cells with acid to mimic the endosomal environment, you can even add in an inhibitor of endosomal maturation, and you can get infection of cells. So we went back to say, yes, that works, but if you do an apples-to-apples quantitative assay, how well does it work? And it turns out that if you take the same amount of virus and add it to cells these two different ways, these are human lung cell lines, that it's about 8 to 10-fold less efficient in terms of the number of cells that come out expressing viral proteins as a marker of successful infection. So the virus is much more efficient at infecting via its physiological pathway than via sort of forcing it to fuse at the cell membrane. And so you can ask, why? Is this a membrane effect, or does this have to do with all the sort of important trafficking that happens in the endocytic pathway, right? Because endosomes travel along microtubules, they recruit a bunch of degradation machinery, and a bunch of this has been shown important to viruses, especially viruses that end up replicating inside the nucleus. Some viruses replicate in the nucleus, some replicate in the cytoplasm. But all this is important to viral infection. So is it really the membrane, or is it all this other stuff? And it's really just a cell trafficking effect. So to test this, we turn to what's really a workhorse technique in our group, which is single virus optical microscopy. And the way this works, I'll tell you about a few variants of it, but we take virus, usually infectious virus, and add it to a microfluidic chamber where we've immobilized target membranes next to a surface. And we can have fluorescent probes in the virus, fluorescent probes in the target membrane. And these probes will change fluorescence to report on changes in viral state, either when the lipid membranes merge or when a nucleic acid binding dye in the target is able to access the viral genome. And so we get images like this, where each spot is an individual virus. And if we take a movie of this, while fusion is being triggered, what you see on a time trace for an individual spot is that it sort of goes along, and then when you get fusion, you get a spike in signal. So either a dequenching spike from the fluorophore or a big quantum gain when the nucleic acid binding dye encounters genome. And so this lets us assess the success or failure of individual viral fusion events and also the kinetics. So we can take hundreds of such time traces and compile them into cumulative distribution functions that show the kinetics and also report a little bit on mechanism. And so that's shown here in red for a lipid mixing dye and in green for one of these genome binding dyes. So this lets us really then assess specifically if we have isolated membranes. Again, if we take these out of cells, then the membranes are no longer metabolically active. You're not worrying about all the other cellular processes and homeostatic mechanisms, and you can really assess sort of apples to apples the effect of different membranes. And so we've done this now for several different types of platforms. We can make synthetic liposomes, shown on the far left, that have a defined composition. We can reconstitute receptors onto those. We can also take plasma membrane vesicles, so essentially induce cells to bleb off bits of their plasma membrane that have shown to be fairly good quantitative representations of the plasma membrane composition. And we can immobilize these and use those as targets. So again, that's a non-metabolically active analog of the plasma membrane. And we've also isolated virus endosome conjugates. So essentially we infect cells, but inhibit the last stage of fusion. We can isolate these conjugates, immobilize these, and then re-trigger fusion in the microfluidic flow cell. So this lets us look at defined composition, plasma membrane composition, endosomal composition. Sort of three different tools and platforms. And what we found initially was that although if you compare liposomes and plasma membrane vesicles and endosomes, the sort of initial stages of fusion look very similar in terms of their kinetics. And that's the sort of lipid mixing between the two. And indeed, all the way through, the kinetics look very similar. But we see for the sort of final stages of pore opening and genome exposure, if we compare plasma membrane vesicles with liposomes, these are synthetic liposomes that are designed to recapitulate the endosomal membrane composition. We again get I think as we do more repeats of this looks like about a 4- to 5-fold difference in infection efficiency. So this is at least half of the effect that we see in cells we can recapitulate with isolated membranes. So this really does suggest that there is a membrane effect. It's not just cell trafficking, and that these different cellular membrane compositions do have a role in controlling the sort of permissive sites of entry. For technical reasons, it's taken us a little bit longer to get the bona fide endosome measurement here because the pore opening assay is a little bit harder to translate to endosomes. That's something we're working on as we speak. But I think this is sort of the key effect. So one of the questions going forward for this piece of the work is to ask whether endosomes are just super permissive to viral entry, or whether different viruses that enter at different sites have essentially adapted to their cellular niche. And so if we look at viruses that fuse at the plasma membrane, Nipah virus is one example. So that's it's a moderate pandemic threat. It's a very lethal but not very widespread bat virus, primarily in Southeast Asia, that infects humans. Most paramyxoviruses fuse at the plasma membrane. And then Viruses that fuse in endosomes include influenza, dengue, chikungunya. Lassa and Ebola tend to fuse a little later in the pathway. And then viruses like herpes simplex virus or SARS-CoV-2 that can fuse in either. You have examples through the evolution of SARS-CoV-2 since its emergence into humans of of strains that fuse primarily at the plasma membrane or strains that fuse primarily in endosomes. So some of these viruses are pretty plastic. And so the question is, now that we've shown for influenza that there are aspects of the membrane in endosomes that are particularly permissive to entry, now when we look at these viruses that might not fuse preferentially in endosomes, we ask, would the endosomal membrane actually be permissive for it, or have they adapted to use compositional aspects of these other membranes more efficiently? And so that's something that we're really interested in to help understand the interplay between the viral fusion machinery and the cellular membranes. Very briefly, one of the things we do in the lab also is molecular simulation. So this is going much, much deeper than the assays, the fluorescent assays that just report on viral state changes. These simulations have one particle for every atom or every 2 to 3 atoms in a reduced model system of the viral fusion protein assembly and liposomes and target membranes. And so this becomes a very computationally intensive endeavor to try to understand molecular mechanisms. So this is an excerpt shown from some of the work where we see a nascent fusion pore between the viral mimetic and the host cell mimetic. And the reason why I'm showing you this is because one of the obvious questions, if we can say there are particular endosomal lipids that increase the efficiency of influenza entry, the obvious question is to say, okay, so now what happens if we put those lipids into these simulations? Can we try to understand why? Because when we start playing with the lipids, we can partially reconstitute it with some chemical analogs, but not fully. And so we want to understand what are the key properties and what are the mechanisms. And the molecular simulation provides a really good way to generate those sorts of explanatory models to correlate with our experimental observations. So switching gears slightly, one of the other key barriers, especially to cross-species adaptation of viruses, are host receptors. And so I've participated in a few sort of international assessment exercises looking at pandemic risk prediction and trying to say, what are the key gaps in understanding to anticipate emergence of new pandemic viruses? And for influenza in particular, there are a number of identified adaptations and barriers, but most people agree that if you had to put your money on something, it would be host receptor recognition. And it's sort of well documented that influenza viruses that infect and transmit between humans and other mammals more efficiently are ones that can bind well to host receptors in the case of influenza in the upper respiratory tract, where then they can be easily infect cells and be processed into respiratory droplets for onward transmission. And so this is something that's sort of a key aspect of adaptation. From a more mechanistic viewpoint, there's the question of, so what role really are host receptors playing in viral entry? They're absolutely key for viral attachment. For some viruses, they've also been shown to mediate activation of these fusion proteins that I said for influenza was mediated by a pH change. For viruses like HIV, host receptor and co-receptor activate the viral glycoproteins as a signal for, okay, now it's time to mediate fusion. Also, spatial organization of the host receptors may help cluster these viral glycoproteins, again, for viruses like HIV that have a sparser array as opposed to a sort of a dense forest of viral proteins to make the process of entry more efficient. And we can test this using a reagent that we've really sort of developed and exploited in a collaboration with Steve Boxer at Stanford. And maybe I'll pause a little bit to describe this reagent because I think it's really cool. It's a fairly simple idea. We take a single strand of DNA and chemically conjugate it to a phospholipid. And these phospholipids will then irreversibly insert into a membrane. So if you incubate just a dilute solution of these DNA lipids with a sample with membranes in it, the phospholipids will insert into the membrane in a more or less irreversible fashion and give you a single-stranded DNA handle. Since DNA hybridizes, if you then have a second treated sample with complementary DNA, you can then tether membrane objects in a programmable fashion. And so we use this, sometimes we use this as a surface immobilization technique, but we can use this in particular as a synthetic attachment factor. So to tether a virus to a target membrane in the absence of physiologic receptor. Other people have used these, for instance, to create artificial cellular junctions. If you're thinking about sort of building tissues or organoids in a synthetic and programmable fashion, this is another tool that people have used to try to couple cells together and create moderately tight junctions between cells in using synthetic reagents. So I think it's something that's kind of cool and has broader utility. But for us, it has this really nice property of we can come in with a membrane, especially a synthetic membrane that we know has no cellular receptors, and we can attach a virus and use that as a way to probe. So what effect does attachment have versus what are the other roles of host receptors? For influenza, these are fusion cumulative distribution functions that we measured using a glycosphingolipid host receptor and using this DNA attachment. And in the case of influenza, really, as best we can tell, cell receptors are just attachment factors. That these curves are identical, and the fusion for immobilized viruses is pretty similar. However, if we compare this for SARS-CoV-2, there's most people now, since it's been, people have heard a lot about SARS-CoV-2. Most people probably know that ACE2 is the host receptor for SARS-CoV-2. And we asked the question of, could we just take this attachment factor and attach SARS-CoV-2 virus-like particles to liposomes, add, instead of pH, SARS-CoV-2 requires a protease. So we add the protease. And is that sufficient for membrane fusion? The answer is yes. But if we compared that to sort of native fusion, so fusion to plasma membrane vesicles from cells that are permissive to SARS-CoV-2 that display both receptor and protease in the membrane, there was a big gap in the kinetics. So essentially, the fusion to these synthetic liposomes with synthetic attachment factors was much slower and actually seemed to, if you sort of analyze the kinetics, requires more rate-limiting steps. So there's a mechanistic difference in the fusion here versus to physiologic target membranes. And so if we add a soluble version of ACE2, so it's not playing an attachment role, but it could play an activation role, then we get a big jump in the kinetics. We don't quite fully reconstitute endogenous kinetics. And we're really interested in this gap. We think these may be spatial organization effects. But this shows that SARS-CoV-2 is somewhat intermediate. It doesn't act like influenza, where the receptors are just attachment factors. The receptors do play an activation role, but that activation role can be spontaneously overcome by thermal fluctuations of the SARS-CoV-2 spike proteins. And so why do we care? We do biophysical chemistry. We love thinking about spontaneous thermal fluctuations and what's energetically accessible. If that's not your thing, why would you care? Because this essentially affects the fitness landscape for adaptation of a sarbecovirus to a new host. So SARS-CoV-2 is a high affinity host receptor. There are a lot of other proteins and especially glycans that have been identified as secondary receptors that are essentially low affinity attachment factors for SARS-CoV-2 on cells. And so if SARS-CoV-2 binds to high affinity receptors that also play an activational role, then there's efficient infection. However, were those absent, it could still infect at a lower efficiency. So if you think about other related viruses that are out there that might not have adapted to use high affinity receptors on human hosts, if they can utilize low affinity receptors, then essentially instead of a very rugged fitness landscape where you have high affinity receptors or nothing, you have a much smoother fitness landscape. So viruses might be able to infect a host, replicate, although not quite as efficiently, and that sort of gives more evolutionary scope for adaptation to use high affinity receptors on that host. So this affects very much our pandemic risk assessment. The other thing, sort of going back to this technology, what it lets us do, and one of the things that we're really interested in doing going forward, is to use this to evaluate pre-pandemic viruses and their fusion to an infection of mammalian and human cells without having to consider gain-of-function genetic manipulation. So say if you have an influenza virus or another potentially risky virus that infects non-human hosts, you're interested in how it might interact with human cells. There's a much debated set of experiments where people can genetically manipulate those to create high-risk viruses to study them. That has a lot of knowledge gain, involves a lot of biosecurity risks. If instead, using these synthetic attachment factors, we can say we have viruses that don't bind human host cells. We can chemically modify them so that this particular virus can bind the cognate cells that we treat, but its progeny won't bind those cells, its progeny won't bind other cells. You have a much lower risk experiment that still lets you assess the activity of these pre-pandemic viruses and also potentially lets you assess candidate countermeasures. There's a lot of work being done on pre-pandemic vaccines, pre-pandemic other antivirals to defend not just against sarbecoviruses, but there's NIAID's doing a sort of prototype pathogen approach where they're identifying key pathogens in different viral families and trying to develop countermeasures. So how do you assess those against potential pre-pandemic viruses, this provides one technology platform. So that's something that we're also very interested in, and something that we have the fundamental technology for, but we really need to do the engineering to implement and scale up, and something that we're looking forward to working on here. So then in my sort of final change of gears, I want to talk about a early stage project in the group. And that's really looking again at this sort of virus membrane interaction. And when the viral spike glycoproteins are activated for entry, they insert, again, you'll recall these short peptides or loops, depending on the viral protein, into the target membrane. These viral membrane-inserted peptides sense their lipid environment and their other properties, and often have conformational switches that can assist with activation. For influenza, that switch is pretty well documented spectroscopically, and that's a pH switch. Even the membrane, not just the whole glycoprotein, but the membrane-inserted portions, undergo conformational equilibria that are modulated by pH and control fusion activity. For HIV, it's a little bit more controversial, but they're pretty clear data showing that there's a cholesterol-mediated conformational switch. So we're interested in whether we can redesign this and also exploit it. So can we take the cholesterol switch and the pH switch and start coming up with mutations that would make, say, an HIV peptide that's pH switched instead of cholesterol switched, or vice versa, a flu peptide that's cholesterol switched instead of pH switched. And talk a little bit about how we're doing this, and then I'll talk about why we might do this. Oops, sorry. So the problem, as I think any of you who have done these endeavors, is that there is a huge combinatorial space of mutations. Even if you have a 2025 amino acid peptide, if you're trying to sort of rationally modify it, that's a lot of potential mutations. Fortunately, and also this is something where it's a little bit hard to do a yeast display or cell surface display selection experiment in this particular case. But fortunately, there's been a lot of work done recently on language modeling. And some of the language modeling work doesn't just come up with chatbots, but has come up with protein language models. And these have also been used successfully on predicting viral mutants. So essentially language models that even without a whole lot of fine tuning seem to do a pretty good job of predicting at least point mutants that are sort of plausibly virus-like. And a number of these come out fairly well in testing against deep scanning mutagenesis experiments. And so this is a logo plot of one of the language model predictions for mutations of the influenza fusion peptide. And our sort of basic first approach is to use these language models as a way to generate a vocabulary. So going from the huge combinatorial space of mutations to a somewhat more limited, but still large space of sort of plausible mutations that keep these peptides in the sort of same local neighborhood of the embedded space, or speaking more colloquially, keep these peptides still looking virus-like. Then, of course, we turn to our other tool that I alluded to, which is molecular dynamic simulation, which can, at the cost of some effort for these membrane-inserted peptides, membrane simulations are, for a number of reasons, much harder than solution phase simulations. But they can capture the conformational changes of these peptides and So we can use simulation as a sort of intermediate screen between the language models generating possibilities and then looking at what are peptides that then might be plausibly switched by the environmental stimuli we're interested in. And then, of course, we can make some peptides and functionally validate them. So both doing sort of basic circular dichroism and other experiments to assess the conformational changes of peptides in response to environmental stimuli, but then there's a solution phase equivalent, a much cruder equivalent of the sort of single virus fluorescence assay that I described to you, where you can look at lipid interchange or content interchange between different liposomal populations driven by viral peptides. And that's been, it's a well-validated assay that's been out there for a couple decades. So the question a little bit is why? If, this is a project that I think we can do, we have some nice initial pieces on this, why bother? I'd say there are two reasons. One is the sort of basic tenet of if we really understand something, we should be able to re-engineer it. So re-engineering as a test of understanding of how these work. So we have these peptide environmental sensors, and if we think we know what the stimuli are and whether they're required, that these are sort of key bits of the viral activation machinery, we should be able to manipulate that. And this is a good way to test, okay, well, what happens if we change this? Does it really have the functional consequence that we think it should? The other thing that's a little bit more forward-looking is if you gave me sort of an oracle, that would be a black box that did this to re-engineer these peptides, what would I want to do with them? If you had peptides that could change conformation in response to different membrane environments, this gives you a very powerful tool to be able to add them to cells and have very local and real-time probes of membrane environment. So if you took like a T cell and were interested in how its membrane environment was changing in response to activation stimuli, checkpoint inhibition, different physiological and pharmacological changes, having these sorts of very localized biosensors would be very useful. So that's a little bit more forward-looking, but why we're interested in this. So broadly, just to sort of zoom out, we're really interested in looking at how emerging, oops, we got a word cut off, but how emerging viruses undergo receptor and membrane recognition and how as they change between host and cellular and intracellular niches, how these two alter. So, for instance, SARS-CoV-2, since the early variants primarily entered via the plasmid membrane, some of the more recent variants seem to primarily enter via mid-endosomes. What permits plasticity in that fashion, or what restricts it? And of course, how can we exploit that? And also, can we, anticipate the factors that are required for new host range? Because one of the key challenges going from here is, you hear about lots of new infections around the world of different sorts. When should you be worried? When should we mobilize resources? A lot of those questions have to do with large-scale population factors, but some of them really can have to do with risk assessment on the subcellular and molecular level. And so we're trying to develop the tools to be able to do that. So with that, I'll stop, and I look forward to any discussion. This is my current team. Underlined are people whose work I've highlighted. Toby is a BME student who's transferring down to tech, and a number of the others are either working at our Sweden site and continuing to do so, or will finish out at UVA and may make some visits. But we'll also have a number of Swedish visitors over the next year as we get up and running. And we'd all love to talk now, talk later, think about new ideas and interactions. And thanks for your time.
Thank you. Bacteriologist here, so might be naive. For your SARS-CoV-2 early and late viruses that change their where they fuse, did you try multiple cell type, like multiple tissue culture cell lines, and do you see the same results?
I haven't. Other people have. A lot of that is driven by their sensitivity to the cell surface protease, TMPRSS2. So the ones that are endosomal fusing are much less sensitive to TMPRSS2. So it's essentially, the let's ignore the membrane viewpoint has been, essentially where's the appropriate protease. What we've shown with some cell-derived membranes is that if you have no protease and then you exogenously add different proteases, the activation mechanisms look identical. So it's just wherever the protease is.
Okay.
We haven't gone and done the experiment of either what you're asking, like endosomes and plasma membrane from 5 different cell lines. But the sort of first order approximation is it's where the proteases are.
Okay.
Yeah, thank you for your talk. It's a really interesting work. I was particularly interested in your DNA ligand binding platform. So, if I recall correctly, you've tried out the situation where the secondary moiety, there's like a library of those being attached to kind of the same receptor that's immobilized. Have you tried kind of the other way around where there's like that immobilized membrane has a library of tethering domains, and then the second moiety is bind to that? And is there any application for that kind of?
Let's see. Let me try to see if I understand your question.
Yeah.
So the way we normally do it is there are two, we use a pair of oligos, of complementary oligos, and then we treat 2 samples with those, and then they'll tether. So you can have it be, one of those is surface immobilized and the other is solution phase. And our collaborators have swapped, so the virus is immobilized, and then there's also solution phase liposomes. We tend not to do a library of the oligos because it requires complementary recognition. What I'd like to use this for, and I think what was sort of schematized in this one, is sort of a one-pot experiment where we have the oligos are all the same, but a library of viruses that are then barcoded separate from this.
Okay.
Does that?
Yeah.
Okay. So I was curious for the single particle experiments, the fluorescent experiments. What temperature did you do those kinetics experiments at? And do you anticipate temperature having an effect? Like lipid composition of the membranes will definitely be affected by temperature.
Yes. We do these at 37.
Okay.
We've done temperature ranges between room temp and 37. For flu, what is tremendously intriguing is you see non-Arrhenius kinetics, that the shape of the CDFs changes with temperature, suggesting that the number of rate-limiting steps changes with temperature. And we can manipulate this a bit, but that's been, for flu, that's been interpreted as the number of engaged hemagglutinin spike proteins that's required, because that's the major rate limiting step for flu, is the recruitment of an engaged spike protein. And so it looks like there's essentially yeah, I didn't include this slide. There's some free energy barrier to membrane fusion. And if you think about, you have the rate of sort of, if you think about a crude model where each engaged spike protein gives you some delta G towards this transition free energy. So you get a bit more, you have a big gap that you could overcome thermally, but it's pretty slow. Get a little bit more, a little bit more. And then of course, when that's enough is going to depend on your ambient temperature. But there's probably flux through all of those pathways at most temperatures, and the max flux pathway is going to depend on temperature. As you say, membrane phase properties are a complicating factor that are also temperature dependent. When we do these experiments, we try to work with lipid mixes that we think are going to be fluid across the temperature range. And that's why we're not going to 4C or 50C.
Hey, Peter, great stuff. Can I just ask you, with apologies to everybody else, explain to me again what the language way of assessing this combinatorial library, this potential library, how does that differ from the ways that people have done it in the past? I don't quite understand.
Yeah, so, oops, where are we? So what this, the way it works, and we can fine tune this more, is essentially you start feeding a model a bunch of, it's first done on like, I think it's UniProt, so lots of proteins, and then you can fine tune it on viral proteins. But you're essentially feeding a model protein sequences and then viral sequences. And it's essentially inferring an embedding into some high dimensional space of these sequences. So you have some mapping from sequence space into some high dimensional space. And the way of generating these candidate mutants is you say, what's something that is a small person who developed this, Brian Haidt, calls it a grammatical distance or grammatical difference, is a small distance in that high dimensional embedded space should be something that still sort of resembles a viral sequence. And so if you do perturb a small distance in that space, run the model backwards, see which sequence that maps to. That's how you can generate the mutations.
But what's the training set data? Like what gets pulled out in terms of to compare to? Are these sequences that are known then to mediate viral entry, or?
Yeah, so the original, so you can do this in two ways. One is the sort of foundational model is just protein sequences, but you can fine tune on viral sequences. And as the training data, the evaluation data, the way that people said, hey, does this work? Is this was also it was a little bit more complicated looking at viral escape mutants. It was to compare to, experiments where people look at, all possible
That's what I was
single
missing.
variant.
Got it. Yeah, thank you.
Good morning. Thank you for the talk. So there was a slide which mentioned that Nipah virus and paramyxoviruses, they take viral entry through plasma membrane, and the influenza, dengue, and the chikungunya, they are following the endosomes pathway. Is there any structural correlation with the viral cellular properties that make them prefer towards such attachment, or like what sort of factors from the viral structural point of view helps them get attached to such cellular structures?
I'm not sure there's a structural rationalization. The simplest rationalization people have is again, and the sort of what I view as sort of the null model from, for some people, this is the interesting part. And for us, this is the null model, is that the entry is wherever the activation factors are present. So some of these, just binding the attachment factor will serve as activation, or attachment factor, and it's a lot of paramyxoviruses are pretty temperature labile. Some things like Ebola require a receptor. So they bind to one receptor at the cell surface and another receptor in the lysosomes. They require a receptor, they require low pH, and they require a protease. It's a lot of stuff. And that's really only present, comes together in the lysosomes. So the main understanding in the field is that the presence of attachment and trigger factors is what drives the site of entry. Our work is sort of saying that if we treat that as sort of first order, what's the second order effect of the membrane? And we see some permissivity effects. And there's a question of, is that also sort of evidence of coevolution to one of these niches? Does that make sense?
Thank you.
There's not an obvious, filamentous viruses always enter here, or, in terms of ultrastructure. That would be lovely if there were a relationship. Anyone else? Great. Oh, yeah.
Sorry, another follow-up
No.
question to the DNA binding experiment. So I guess outside the context of viral infection studies, has the same platform been used to study, like, I don't know, like cancer metastasis or like environmental biosensing, those kind of different it's pretty limited.
Let's see. The major things I'm aware of are using this one for, you can sort of, you can have then tethered but mobile liposomes, and people have sort of set this up to be able to do like essentially electrophoresis, right? If you apply an electric field, you can sort populations. People have used it for artificial cell-cell junctions and organoids. That's mostly Zev Bryant. There's a similar technology using, instead of phospholipids, using sterols that go in more easily, but also it's reversible. And people have used that for some other design applications. I'm not aware of any cancer work, but especially on the sterile DNA, there's been a little bit more, and I don't know everything in the field.
Thank you.
Great.